Hello students, and once again welcome to the program of Bachelor of Science 3rd year, subject Botany, semester 5, course code BOD 101 and course title Plant Tissue Culture. Title of the Unit 4, Types of Cultures. Module name is Principle, Protocol and Applications of Meristem Culture. In this module, I'll be stating the principle of meristem culture, explaining protocol of meristem culture, and lastly, listing and explaining the applications of meristem culture. At the end of the same module, student defines meristem culture, states the principle of meristem culture, explains protocol of meristem culture and explains the applications of meristem culture. Now, what is meristem culture? It is the in vitro culture of apical meristem which is measuring less than 0.1 mm together with 1 to 3 leaf primordia under aseptic and controlled conditions. Towards the right side, you can see the meristem and the shoot apex. Both of them can be grown in vitro into the plantlet. Now, what is the principle of meristem culture? It is the excised shoot tips and meristem that can be cultured aseptically on nutrient medium under controlled conditions to grow directly into a small leafy shoot. So, towards the right side, you can see the culture tubes. These culture tubes are with the media, and in each of the culture tube, in one tube, the meristem has been inoculated, whereas in the other tube, the shoot apex has been inoculated. So, once these meristem and the shoot apex, they are incubated aseptically under controlled conditions, they are going to grow directly into a small leafy shoot. Now, the protocol. The first thing is excise the tip, which is 1 centimeter portion of the young twig. Surface sterilize the tip with 2% sodium hypochlorite solution for 10 minutes. It's very important to surface sterilize with the sterilizing agents because these sterilizing agents are going to kill the microorganisms that are present on the surface of the explant. After 10 minutes, rinse thoroughly with sterile distilled water thrice, again to remove the sterilizing agents, because sterilizing agents, if they remain on the surface of the explant, they will be very harmful for the growth of the explant. Later, transfer the tip in the sterile Petri plate. Now, excise the meristem, which is of 0.1 mm size, with the help of a scalpel and transfer to the nutrient medium, which will be MS medium, which is also called as the Murashi's and Skook's medium. Now, once the meristem is transferred to the nutrient medium, the incubation has to be done. So, incubate the culture at the temperature of 25 plus minus 2 degrees and 16 hours photo period. So, when the cultures are incubated aseptically and under controlled conditions which have been mentioned over here, they will develop into a small leafy shoot. Now, coming to the number of applications. As you all know that meristem is consisting of the meristematic tissue in turn contains meristematic cells. And all these meristematic cells, they are virus free. So whenever we grow a leafy shoot from a meristem, naturally all these shoots which are grown, they will be virus free. So the plantlets that are grown from the meristem or through the meristem culture, they are virus free plantlets. The second application is the micropropagation. That is, you can grow number of small shoots, and quite a number of small shoots can be grown in a tube. The third one is used in plant breeding. Now, during the plant breeding experiments, the number of hybrids are formed, and these particular hybrids, they can produce the recalcitrant seeds, that is, the seeds which do not germinate. So these hybrids we can use to grow into number of plantlets through the meristem culture. Lastly, the storage of genetic resources, that is, certain seeds which cannot germinate, or the seeds which cannot be stored for a longer period of time. These seeds cannot be stored as a genetic resources, or their storage for genetic resources cannot be done. So, these particular meristem culture can come to the rescue, and we can store the meristem as a genetic resource. So these were the few of the applications of the meristem culture. Now, when we look at the applications of the meristem culture, we immediately come to know the importance of the meristem culture, mainly because the plantlets that we are going to grow or develop through the meristem culture, they are going to be virus-free plantlets. And another important thing is you can store this meristem as a genetic resource. These are the few of the references that were used for the e-module or for preparing this e-module. Thank you.